Hello, everyone, and welcome to today's webinar, The Evolution of Precision Diagnostics and Advancing Treatment Strategies of Solid Tumors. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Covaris. To learn more, please visit covaris.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to welcome our speakers, Eugenio Daviso, Vice President of Solutions at Covaris, and Jeffrey Conroy, Head of Science at OmniSeq, Executive Director at LabCorp. Eugenio, I'll hand it over to you to get us started.
Thank you for the wonderful introduction. My name is Eugenio Daviso. I'm the Vice President of Solution at Covaris. And today I would like to share a story that is not just about Covaris and our accomplishment in sample preparations. Instead, it's about why we as a team are dedicated to solving the challenges that reference and clinical labs are facing with nucleic acid extraction in oncology. It is about our commitment to advancing the fight against cancer collaboratively across organizations with the help of our adaptive focused acoustics or AFA technology and through extract FAP smart solutions. Over the past 25 years, Covaris has become a trusted name in the field of DNA sharing, providing tailored solutions that cater to next-generation sequencing workflow for both germline and somatic analysis. We've been proud to assist laboratories all over the world in scaling their operations, whatever that means going from low to high throughput or integrating automation solution into their processes. In the last 5 years, we've seen exciting advancement in our field by developing focused acoustic ultrasonication methods combined with a tailored buffer composition, thus acting on the chemistry. What we find remarkable about this combined technology, it is its versatility. It does so much more than just creating shearing forces that fragment the nucleic acid. It can also promote molecular processes like trypsin digestion, mixing without disrupting biological system, and it can even lyse cells from various sources, such as cultured cells, frozen tissues, or formalin-fixed paraffin-embedded tissue, providing high-quality nucleic acid, proteins, metabolites, or other biomolecules. At Covaris, our vision goes beyond the here and now. We are working to automate workflow and standardize sample preparation performance across the board. We believe in the value of high-quality biomolecules since they hold immense potential for downstream analysis, whether that is through nucleic acid sequencing, mass spectrometry, as well as other methods like PCR, fluorescence, or immunoassay. Together, with our customers and collaborators, we are pushing the boundaries on what is possible in the fight against cancer in a collaborative way. To date, the FDA has approved about 140 nucleic acid-based companion diagnostic devices. Among these, 70 of them are tests specifically dedicated to the field of oncology. This underscores the crucial role and potential of nucleic acid-based diagnostic in cancer care and treatment. 10 tumor profiling tests may not seem like a lot, but we should consider that just 5 years ago, in 2018, only one such test, the MSK Impact, was available. From this test, two main applications can be considered mainstream today: molecular residual disease and comprehensive genomic profiling. This headline at the bottom of the slide is from the Memorial Sloan Kettering Cancer Center website, and it captured the heart of our mission and the reason why we are so passionate about what we do. It is a powerful statement to the possibilities of current genomic profiling methods to transform cancer treatment. The ability to cure 10 patients, as stated in the article, it is not just a statistic or a milestone. It represents real individuals, real lives that have been impacted by the collective effort within the field of genomics. It is stories like this that are inspiring us and driving us to deliver a robust solution to the root of the comprehensive genomic profiling workflows, which is sample prep. Our FAP Smart solution was designed with a benchmark NGS workflow in mind, that is the TrueSight Oncology or TSO 500, tested by Illumina. This test, which holds a CE mark, offers comprehensive genomic profiling. Notably, the TSO 500 test aims to match cancer patients who have rare genetic mutation with a targeted treatment. TrueSight Oncology 500 targets 523 genes for DNA analysis and 55 genes for RNA analysis. In total, the test produces 7 different biomarkers covering all variant types. Our FAP Smart solution seeks to enhance this process, improving ease of workflow, yield, and quality of nucleic acid extracted for such analysis. In discussion with experts from NGS and oncology labs, we have come to understand the main challenges presented by FAP tissue. These are ranging from the labor-intensive manual deparaffinization process to the often insufficient quantities of extracted nucleic acid, which are frequency or poor quality. These difficulties underscore the need for greater yield with minimal tissue input, as well as simplified and automated workflows. Additionally, labs require suitable vessel design for effective tracking and easy accessioning. The ultimate goal for this lab is to achieve actionable outcomes, whether that is identifying an effective treatment or qualifying patients for clinical trials. Since 2017, we launched several kits that are supporting different batch sizes with both a manual or semi-automated workflow, such as the solution implemented by OmniSeq that will be featured in the next presentation as a part of their Insight workflow. And these kits are captured on the center left of the slide. We recently launched the TrueExtract FAP Smart solution by enabling flexible batches and throughput by designing its workflow around full automation, as well as semi-automated modular solutions from deparaffinization to purification. Our solution goes beyond simply addressing the technical challenges of nucleic acid extraction and purity. We also offer capabilities for DNA sharing, which is a necessary step for the TSO 500 test. Our goal is to establish collaborative relationship with laboratories performing comprehensive genomic profiling. By working together, we can contribute to generate more and more reliable and accurate sequencing results, ultimately enhancing the value and impact for these vital tests. These slides provide an overview of our FAP Smart solution features and benefits. It also highlights on the right side of the slide the 3 configurations that are tailored to meet different lab needs, from fully automated workflow utilizing the Hamilton Star, which is a project we are currently developing in collaboration with Hamilton and OmniSeq, to modular semi-automated solution using dynamic devices or Hamilton liquid handlers and deep purification system. While the semi-automated solution do involve more touch points, each configuration is designed to streamline the process and is adaptable to the unique requirement of each lab. But the benefit of our solution aren't just technical. We are also focused on the practical advantages that directly impact lab operations. The solution is designed to save time and reduce cost while offering flexibility to minimize or adjust the desired level of hands-on time. Furthermore, our solution enhances the accuracy and precision of NGS outcomes. This is our commitment, not to only increase the efficiency of genomic profiling, but also to improve its reliability significantly. This slide is a summary of the performance metric for both the DNA and RNA of the TSO500 test, which were collected from a diverse population on over 170 FAP samples using our solution. These samples are from a variety of tissue types and were obtained using minimal input methods, such as fine needle biopsies or slide scraping. Using our unique chemistry, we are achieving very promising results. More than 90% pass rate for the DNA matrix, which include single nucleotide variants, tumor mutation burden, copy number variation, microsatellite instability, and contamination matrix. Moreover, our pass rate currently for RNA is 100%. These metrics are specifically outlined in the TSO500 manual, and the results demonstrate to us the high level of reliability and accuracy that our solution can offer, even under challenging conditions. I'd like to extend my deepest gratitude to the OmniSeq team for their incredible support and for aiding us in understanding the need of a state-of-the-art clinical lab that constantly strives to improve patient outcomes. Their insight into achieving more reliable results and faster turnaround time have been invaluable. I also want to acknowledge the Covaris team who have worked tirelessly and continue to do so to create a new paradigm in the field of sample preparation for oncology. Thank you all for your attention and for joining us in this journey toward advancing cancer diagnostic and treatment. Now it is my distinct pleasure to introduce Jeff Conroy, the Head of Science at OmniSeq LabCorp. Jeff and his team have been instrumental in developing and deploying remarkable advancement in the field of oncology over the past few years. I'm confident that you will find this presentation enlightening as he shares with us their pioneering work. Please join me in welcoming Jeff.
Thank you very much, Eugenio. And my talk is also a story on progressing precision diagnostics in support of precision medicine and the role that industry partners such as Covaris have played in advancing tumor profiling. As a quick introduction, I'm head of science at OmniSeq, located in Buffalo, New York, where we provide comprehensive genomic and immune profiling to support clinical testing and biopharma services through LabCorp Oncology. All testing is performed in our CAP-CLIA lab using New York State CLEP-approved assays developed under ISO 1345 quality management system for med devices. Our first assay, OmniSeq Target, launched in 2014. So we've been active as a clinically approved NGS lab for nearly a decade, first as part of the Roswell Park Comprehensive Cancer Center, then as the OmniSeq spin out in 2015, and now as part of the LabCorp Oncology since our acquisition in 2021. So, as you know, healthcare, in particular oncology, is undergoing a transformation with the promise of personalized medicine and precision diagnostics changing the way we diagnose and treat disease, leading to a more precise, predictable, and customized approaches for the individual patient. And this is all based on their own unique genomic makeup. And if we are to fulfill our vision of bringing the promise of precision medicine for cancer care within reach for all, test developers, manufacturers must lead the way in leveraging innovative technologies to detect unique cancer-causing alterations in a robust, efficient, affordable, and compliant manner, and as importantly, enable clinicians to quickly and accurately choose the most appropriate course of action for a patient. What may seem as a surprise to many, most oncology patients are treated outside the academic centers that run clinical trials and are considered the pillars in precision medicine initiatives in support of drug development and patient recruitment. Upwards of 85% of cancer patients are treated by community providers geographically scattered throughout the US and distant from these major academic institutions. With an estimated 80% of patients not receiving sufficient testing aligning to FDA-approved targeted therapies or clinical trials, there is an urgent need to improve access and delivery of personalized medicine approaches. So in this talk today, we're going to focus on solid tumors and describe first the inherent challenges in providing precision treatment options for cancer patients based on tissue profiling, and then provide an overview of comprehensive genomic and immune profiling and its impact on therapy selection. Discuss some recent innovations that have led to broad-based NGS assays becoming standard of care. And lastly, we'll conclude with a review of the therapeutic actionability of 10,000 cases which recently underwent comprehensive genomic and immune profiling using our OmniSeq Insight test. So to set the stage a little, what is precision medicine, right? In short, it aims to match patients with appropriate molecularly matched treatments based on their unique tumor profile. And the tumor profiling is used to identify biomarkers, which include somatic mutations in DNA, variations in copy number, perturbations in gene expression and protein expression. And in total, identification of key biomarkers can inform clinicians and researchers of patient cohorts who may best respond to targeted therapies designed for their specific alteration, advance correlative science efforts, and stratify patients to enrich for clinical trials. For decades, many cancers were treated similarly with surgery, radiation therapy, chemotherapy, combinations of both. But more recently, identifying biomarkers in cancer cells or cells in the tumor microenvironment has led to the development of new precision medicine drugs, such as targeted therapies and immunotherapies, designed to target specific features in cancer cells, potentially reducing the damage to healthy cells. The routine use of a variety of biomarkers has substantially changed the way in which cancer medicine is practiced, from providing more accurate prognostic information to assisting in the prediction of specific therapeutic strategies that are more likely to result in a favorable outcome for an individual patient. As you can see here, there have been several biomarker-specific solid tumor approvals in the United States and the European Union as examples of the rapid advance in the number of available biomarker-driven treatment indications across multiple tumor types. And this only includes approvals related to single-gene genomic biomarkers, not multi-gene testing or immunohistochemistry assays. But multi-gene testing is also critical for precision medicine, as represented here by homologous recombination deficiency. Alterations in any of these HRR genes, which are several, reduces the cell's ability to repair DNA damage, leading to the HRD phenotype of genomic instability as measured by large-scale transitions, telomere allelic imbalances, and loss of heterozygosity. NCCN guideline recommends the testing of genes associated with hereditary breast, ovarian, pancreatic, and prostate cancer, and germline and/or tumor testing for alterations in HR genes or HRD status in ovarian cancer. And recent clinical evidence has shown that patients identified with HRD by molecular testing have improved survival when treated with a class of drugs called PARP inhibitors. As with HRD, the detection of MSI and high TMB requires multi-gene, multi-loci testing to determine the status of these primary immune markers. Each assay surveys large regions of the tumor genome to measure the level of genomic instability as a surrogate for neoantigenicity, which allows both to serve as a predictive biomarker of response to immune checkpoint inhibitors in several tumor types. Two pivotal studies have demonstrated improved progression-free survival in MSI and TMB high cell tumors treated with pembrolizumab, which is an immune checkpoint inhibitor targeting PD-L1. But genomic biomarkers are not the only markers of response in solid tumors. The immunosuppressive tumor microenvironment is a major component of resistance to immune checkpoint inhibitors, and there are several immune markers, in addition to PD-L1, when elevated, have been associated with improved response to checkpoint inhibitor therapy across multiple histologies. Some of these markers are expressed on tumor cells, but the majority are expressed in T cells, antigen-presenting cells, and other cell types that make up the tumor microenvironment. Emerging therapies for several of these secondary immune markers target alternate checkpoint pathways outside the PD-1, PD-L1 axis, and also immunomodulatory genes associated with T-cell activity. As an example, Thulog was recently approved for treatment of advanced melanoma by targeting both LAG-3 and PD-1, with several other tumor types being studied in clinical trials, highlighting drug development efforts to assess a new class of immune checkpoint inhibitor treatment with more in the pipeline, including cell-based therapies and cancer vaccines. So as you can see, this ever-changing but promising biomarker landscape makes it challenging to not only keep up with the new drug indications, but also the associated biomarkers and testing required. But these aren't the only challenges. What are options for patients that are negative for the three primary immunotherapy biomarkers, PD-L1, MSI, and TMB, and those that are also negative for markers associated with targeted therapies? How do we extract as much information from the tumor biopsy, which can be extremely small and the only specimen available? To make precision medicine work, complex, technologically advanced testing and workflows are required to meet these real-world challenges that we deal with every day. So let's start with the samples. As you can see, they come in all different shapes and sizes. Tissues are collected through invasive procedures and preserved in formalin fixative. It is ideal for maintaining cellular morphology and preserves the sample for staining and microscopic review, but is not ideal for preserving the DNA and RNA molecules used for molecular diagnostics. So from the very beginning, challenges in the use of tissue preserved in formalin fixative needs to be overcome, not to mention the heterogeneous nature in which tissue is procured in pathology labs across the country. So across all these different specimen characteristics, there's a need to have standardized methods to isolate the valuable nucleic acid for downstream testing. When precision medicine first emerged, there were only a handful of therapeutically matched biomarkers that required testing, and they were typically performed on a single tumor type or a single gene using conventional FISH, immunohistochemistry, qPCR, or Sanger sequencing methods. The testing was limited to markers with approved therapies and was very limited in scale and result reporting. And this presented challenges to testing laboratories, as each one of these single gene assays required their own supportive platform, each with their own method, training, operator competency, and reporting requirements. And this was most apparent with non-small cell lung cancer, which currently has multiple biomarkers recommended by NCCN for testing. Using a traditional single gene testing approach, over 30 slides is required to meet the testing guidelines. These are typically performed iteratively, waiting for results of one test before starting another, and can extend testing, for several weeks, and often result in the consumption of precious tumor material prior to testing all the recommended biomarkers. So how do we solve the simultaneous challenge of a difficult starting sample, the need to test multiple biomarkers that have limited tissues, the need for potentially multiple platforms, the need to perform therapy trial matching, provide a concise standard report for a physician, and do this all in a clinical actionable turnaround time, and to do it at scale so we can reach everyone. We think that the solution that's implemented at OmniSeq and LabCorp is a comprehensive genomic and immune profiling assay, which is a collection of innovative technologies that allow the testing of multiple biomarkers from minimal specimens, resulting in a single report of relevant detected variants and associated therapies. Comprehensive genome immune profiling takes advantage of innovations, including next-generation sequencing, to provide specialized assays to perform both genomic and immune profiling, with the genomic profiling focusing on tumor-specific alterations associated with response to targeted therapies, and immune profiling designed to understand the host immune response to cancer. Corresponding workflows and pipelines have been designed to allow the simultaneous evaluation of both genomic and immune profiling from a single sample to support treatment and clinical trial decisions. These assays can target hundreds of genes at a time and report alterations at the single nucleotide level, exon level, gene level, and chromosome level, including single nucleotide variants, copy number variation, gene rearrangements, splice variants, TMB, MSI, and regions of loss of heterozygosity. Furthermore, quality management systems such as ISO 13485 for med devices and FDA 21 CFR Part 820, which provides foundational CGMP regulations, are available and are utilized to ensure that the systems and processes used in the design and manufacturing of these assays meet customer and regulatory requirements and, of most important, accurate reporting. And this includes a number of tumor profiling devices that have CE marking and FDA approvals as companion diagnostics. So what are some of these innovations that have led to successful high throughput comprehensive genome and immune profiling as a precision diagnostic assay? If we break the CGIP workflow down, there's 6 major components that are grouped into pre-analytical workflow, the analytical workflow, and the post-analytical workflow. The pre-analytical workflow starts with the receipt of FFPE tumor specimens and the anatomic pathologist tissue QC review and the extraction of DNA and RNA from the tumor-enriched sections of the specimen. Following DNA RNA QC and normalization, the analytical workflow begins with library preparation. And in this step, it targets and enriches for genes of interest and creates sequence-ready libraries that are individually barcoded for multiplex testing and patient assignment. The analytical steps are completed once the individual patient libraries are pooled together and sequenced. And then the post-analytical workflow begins with raw sequencing data that moves through a bioinformatic pipeline to perform variant calling with subsequent annotation and therapy trial matching. The workflow ends with review and sign out of a clinical report by a board-certified pathologist. So how do we effectively track and move hundreds of samples per week through this complex workflow and maintain a 1-week turnaround time from sample receipt to report? The first thing to point out are the availability of supportive IT systems that have been designed to manage the complex testing workflow and data workflow from order entry through reporting. These systems handle pre-analytical, analytical, and post-analytical processes through an integrated set of software tools to ensure sample tracking, identity, preparation, sequencing, analysis, and report sign-out. When built in a compliant fashion, these systems provide a hands-free environment that ushers through hundreds of samples at a time with auto-generated work lists, barcoded IDs, sample sheets, and instrument APIs that track and qualify samples for the entire process. These systems are imperative to meet a 1-week turnaround time from sample receipt to report and facilitate the integration of cancer genomic data into EHRs to support clinical decision-making. So now let's take a look at the sample and associated library preparation workflows. NGS technologies that support comprehensive tumor profiling have advanced to require as few as 5 unstained slides or DNA RNA inputs as low as 10 nanograms, depending on the assay used. And from this minimal input, hundreds of actionable mutations can be evaluated, including all Tier 1 mutations with FDA approvals or in NCCN guidelines. Most are validated for use with all solid tumor FFPE tissues, including resections, needle core biopsies, and fine needle aspirates, which are the most common methods of obtaining tumor biopsies. And although FFPE tissues are not ideally suited for molecular testing, the samples are remarkably stable in ambient temperatures and can easily be placed in transport boxes and shipped to central laboratories by a number of overnight carriers. After tissue QC, tissue sectioning, and the scraping of the tumor-enriched areas off the slides, a variety of methods, chemistries exist that allow DNA and RNA to be co-extracted and are of sufficient quality and quantity for the downstream applications, including comprehensive tumor profiling. One such workflow that Eugenio highlighted in the introduction is the Covaris true extract method, which we use at OmniSeq. The workflow uses individually 2 D-bottom barcoded tubes that are 96 well compatible and minimizes variability across samples in a mostly hands-off process. The acoustic focus technology that Eugenio mentioned is a key component in the deparaffинization steps, where the Covaris process eliminates the need for xylene-based paraffin removal and reduces the impact of high wax tissue ratios in certain samples on downstream processing. Secondly, using either a filter or a magnetic bead-based purification workflow, we have demonstrated, as you saw, that the true extract they have a higher tolerance for paraffin and non-soluble contaminants compared to other column membrane-based methods, and overall higher yields, especially with smaller tumor samples. And most importantly, as you saw earlier too, the extracted DNA and RNA performs exceptionally well across a number of downstream applications, including next-generation sequencing. And finally, as seen here, the workflow is specifically developed for full automation as an assay-ready workstation, as with our collaboration with Covaris and Hamilton. Our system that you see here is designed for 16 to 96 sample throughput in around 8 hours, with only 4 manual touch points during the extraction process. Another key component of the pre-analytic workflow is shearing the DNA extracted from the formalin-fixed paraffin-embedded tissues to a uniform size for efficient library preparation. Once again, the Covaris AFA technology is the recommended solution for several NGS-based methods, as it uses finely tuned acoustics to achieve consistently sized, randomly fragmented DNA. As with the true extract workflow, the system is automation friendly, enables high throughput preparation for a full plate of 96 samples with reproducible and repeatable results regardless of the inherent specimen characteristics, tissue mass, and paraffin amounts. Moving on to the analytical workflow, advancements in library preparation and next-generation sequencing have enabled comprehensive profiling to be cost-effective by targeting regions of interest and maximizing the number of samples that can be pooled in a single batch. And as a reminder, there are numerous NGS applications for which a few related to human health are depicted here. For RNA, as an example, transcriptomic sequencing has advanced oncology testing by detecting expressed fusions, splice variants, and immune gene markers that are associated with therapy response, and is also being used in studies demonstrating different signatures that can predict patient response to immune checkpoint therapies. Focusing on DNA, methodologies have evolved to allow sequencing of the whole genome, or a focused approach on exonic regions, or targeting specific regions such as actionable mutations or genes in the drug development pipeline that are most relevant to the test's intended use. Targeted approaches that use either amplicon or hybridization-based capture methods in enriching target sequences associated with oncogenes and tumor suppressor genes have brought a cost-effective option to laboratory testing of solid tumors. A technological advance that has especially helped with rare variant detection from low amounts of starting FFPE-derived material is the use of unique molecular indexes, or UMIs. Molecular identifiers have been given various names, but they are usually designed as a string of random nucleotides ligated to individual DNA molecules or through primers during PCR. And UMIs that are added to sequencing libraries enable downstream bioinformatic identification of PCR duplicates and sequencing errors from matched reads. UMIs also help with error correction by correcting DNA damage induced artifacts of the formalin fixation process by comparing the sequences from each strand. And together, the targeted sequencing approach combined with error correction capabilities increases the accuracy of the sequencing data significantly, allowing high specificity for small variant calling and TMB calculations. As shown on the right side, a true mutation, illustrated as a red star, is expected to be present in all the reads carrying the same template molecule tagged with UMI, while an error, illustrated as a blue star, is expected in some but not all the reads carrying the same UMI. UMI reads are collapsed into families where discrepant calls are removed, and then a consensus sequence is called. One such example of a targeted sequencing assay that uses both DNA and RNA as inputs is OmniSeq Insight. OmniSeq Insight was developed in our laboratory under CAP/CLIA guidelines and ISO 1345 design control, and was launched in 2021 following New York State CLEP approval. The test uses PD-L1 immunohistochemistry, the Illumina TSO 500 assay for genomic profiling of 523 genes, and the Thermo Fisher Oncomine immune response assay for the immune profiling of 395 genes. Altogether, this test targets over 900 genes to detect small variants, copy number, fusion, splice variants, HLA class 1 haplotypes, TMB, MSI, and other markers associated with immune checkpoint pathways, cytokine signaling, T-cell activity, cancer testis antigens, and other markers associated with targeted therapy and immunotherapy. The content of these assays were developed with pharma to align with drug development targets and therapies in the oncology pipeline. And as I will show later, this assay has been critical in matching patients to approved therapies and also to ongoing clinical trials based on their genetic makeup. However, without innovations in liquid handling and downstream sequencing systems, testing patients in an efficient manner would not be possible. So several automation vendors have developed and validated library preparation workflows that not only cut down on manual processes and operator fatigue, but also minimizes variability across and within batches and facilitates tracking of samples and reagents in this complex workflow. Likewise, sequencing vendors have developed benchtop and high-capacity instruments that can meet the demands of laboratories performing tumor profiling. With faster runtimes, improved sequencing quality, higher capacity, and lower costs, NGS has enabled comprehensive genomic and immune profiling to become the standard for which patients and treating physicians can depend. So moving on to the post-analytical workflow, which includes pipeline analysis and the sequencing results, variant calling, quality control measurements, annotation, variant interpretation, reporting. A number of systems here also have evolved to take the raw sequencing data for hundreds of libraries and distill them down to a concise report within a few hours. Bioinformatic pipelines have been developed to automatically receive messages from the sequencers to begin analysis as soon as the run is completed. Automated scripting and monitoring occurs as the analysis gets split between nodes of high-performance computing clusters, specifically designed to demultiplex the sequence libraries and assign each to an individual's barcode, perform the variant calling across all the variant classes, and then communicate success or failure across all components. These systems can be on-prem or hosted in a cloud environment to allow scale and quick node rebalance for fast turnaround time. Our system can process 144 libraries in less than 12 hours from sequencing completion, delivering qualified small variant TMB, MSI, copy number, HLA fusion, splice variant, and gene expression results as distinct run message deliverables. These run message deliverables, along with sequencer and sample level QC metrics, are then pushed to a laboratory information system for review and finalization of reporting. Specialized list systems like the Genome Oncology Workbench that we use and others have been designed to take sequencing data and merge the patient, demographic, order information, and a functional user interface. Here, rules are applied to allow variant level reporting of those passing analytical quality thresholds, with rule-based therapy and clinical trial matching and selection occurring following the latest standards and guidelines of somatic variant interpretation and reporting. These systems allow for the objective and consistent sign-out of reports between pathologists and truly personalize the reports by matching therapies and trial that are specific to the patient's tumor type and detected alterations. Furthermore, multi-marker matching is enabled by list functionality to facilitate clinical decision-making for patients with multiple markers that may be detected and considered actionable for both targeted and immunotherapies. Once the report is generated, reviewed, and signed out in the list, the system will automatically distribute the reports, update the order status, and move data to secure databases for archiving. As mentioned, the list report generator takes only a few seconds to create a patient report. So from hundreds of genes and the sequencing of millions of base pairs of DNA and RNA, report can be condensed and limited to those markers that are considered to be clinically relevant. The first page has all the patient, specimen, and client information, along with the marker findings and any testing comments. The second page associates the marker findings listed on the first page with therapy considerations, splitting them into those which have clinical benefit in the patient's tumor type and those with emerging clinical benefit as approved by FDA or recommended in NCCN guidelines. The latter pages of the report then provide clinical trial options, again based on the markers detected in the patient's tumor. As mentioned previously, most cancer patients are treated in the community setting, so these reports can prioritize trials based on proximity to the patient's home address to help bring these new therapeutic options to the patient. So what is the impact of comprehensive genome and immune profiling on real-world cancer patients? Looking at 10,000 cases tested with OmniSeq Insight, the majority of tumor types tested are lung, followed by colorectal, breast, cancers of unknown primary, pancreatic, prostate, esophageal. This makes up about 75% of the tested population and accounts for only 7 tumor types. The remaining percentages represents 28 tumor types, highlighting utility of comprehensive tumor profiling as a pan-cancer assay. Patients tested with OmniSeq Insight come from 44 states and highlights the ability to provide broad access to cutting-edge testing at our Buffalo, New York lab through the LabCorp distribution network. However, it also underscores that there's more work to be done in providing access to regions not depicted here. And most importantly for patients, when looking at turnaround time over the past year for OmniSeq Insight, the average turnaround time from sample receipt to clinical report is just over 7 days, highlighting that all the systems and technologies that we've discussed have evolved to process and report hundreds of samples per week in this clinically actionable turnaround time. Looking at cases from a genomic alteration viewpoint, we see that single nucleotide variants depicted here as dark blue and copy number variants as yellow are the most prevalent alteration types detected. This is not surprising since the number of potential alteration positions is highest for small nucleotide variants, but sprinkled in are a number of genes with higher rates of fusions here in light blue. TP53 is the most frequently mutated gene, followed by LRP1B, SBTA1, and ZFHX3, all driven by small variant alterations. KRAS and ATM follow this grouping with both CNV and SNV driven alterations. Looking at other CNV driven genes, PIK3CA, P10, BRCA2, and EGFR have copy number change frequencies approaching 50%. If we distill all detected mutations to those that are considered matchable to either a targeted therapy and or immunotherapy and guidelines on label or associated with clinical trials, and group them into alteration types of SNV indels, copy number gains, loss, fusion, splice variants, etc. We see that anywhere from 1.6 to 95% of patients have a biomarker that is considered positive across any single class of alteration type. The immune markers and SNV indel alteration types have the most positive markers with copy loss, splice variants that include exon 14 skip and EGFR V3, and MSI high having the lowest percentage of positive patients. If we look at the alteration types further and classify the matchable alterations into tier 1, which are on label or in NCCN guidelines, we see that nearly all MSI and TMB high patients match to an approved therapy, which is not surprising given that these are tumor agnostic markers for immune checkpoint inhibitor therapies. Then if you look at the tier 2 matchable alterations, which are those that are eligible for clinical trials, a high percentage of patients that do not have tier 1 alteration do have alterations that match inclusion criteria for at least one trial. Clinical trial matching is driven by copy number variations, fusions, and immune markers in drug development. This subset of patients would only have a trial available to them, with several driven by off-label studies. For splice variants, EGFR V3 is the predominant splice variant for trial matches. And finally, those alterations that do not match to either a non-label therapy or NCCN guidelines, nor match the clinical trial, you see that the number of patients that do not match either category are minimal, with the most common alteration type not matching being the immune markers, for which several are still in early development, and as of the date of the report sign out, did not have a biomarker with enough clinical evidence to support a match for either a drug or a trial. When looking at the matchable tier 1 alterations across the 7 most common tumor types tested, and again, we define tier 1 alterations as those on label or in NCCN guidelines for either targeted or immunotherapies, we see that nearly 85% of lung cancer patients tested with OmniSeq Insight had a tier 1 alteration detected. In some ways, this seems high, but when you consider the number of biomarkers associated with personalized therapies and the prevalence of positivity, for example, greater than 50% are PD-L1 positive at the greater than 1% TPS cutoff for 22C3, and likewise upwards of 50% are positive for either an EGFR or KRAS mutation. The power of the combined approach of simultaneously testing all relevant biomarkers at one time becomes apparent. Focusing again on lung and recalling the earlier slide that highlighted all the single gene testing required to match patients with appropriate targeted therapies, this figure is a snapshot of the 4,000 plus lung cases in this study, the detected matchable alterations, and the targeted therapies available and matched by OmniSeq Insight. Again, emphasizes the need to stay current with the ever-changing guidelines and biomarkers, match the tumor alterations based on guidelines, and accurately generate a report. Recent studies looking at real-world data for newly diagnosed stage 4 non-small cell lung cancer patients found outcomes were significantly compromised if actionable mutations were identified after treatment, such as chemotherapy and immunotherapy, was initiated. But the good news is, when treatment was initiated based on molecular results, patients experienced better outcomes. Just a quick review of the other tumor types that make up the 25% of the cases tested. We find that cervical cancer, melanoma, head and neck, bladder, thyroid, stomach have the highest rate of therapy associations, approaching nearly 50% of patients and higher. Many of these are due to the high PD-L1 positivity rates, especially seen in cervical cancer and head and neck cancer. But some, like melanoma, are driven by BRAF mutations, stomach cancer by ERBB2 amplifications, and so on. Lastly, there are a number of tumors that historically have a low rate of FDA and NCCN therapy associations. The list here shows those with less than 25% patient positivity and includes rare tumors such as sarcoma, liver, kidney, gallbladder, and central nervous system tumors. So in summary, we've come a long way since 2014 when we launched OmniSeq Target, which was a 23 gene assay that at the time required next generation sequencing, FISH, and other technologies to detect all the different variant classes and markers. We could run 8 samples per run, about 24 per week, and it took nearly 2 weeks to report. And at the time, the industry was lacking consensus somatic variant interpretation and reporting guidelines, making standardizing the report a real challenge. However, things have changed, and we and others have shown that it is possible to bring together innovative technologies in sample transport, sample processing, library chemistries, next-gen sequencing, and supportive software tools to create a scalable, high-throughput, multi-omic assay for comprehensive tumor profiling that in our lab can run 70 samples per batch and hundreds per week. And by maximizing tissue utilization and resultant biomarker information through multimodal, broad panel, comprehensive profiling, we can provide a more complete picture of the tumor and bring the promise of precision medicine for cancer care within reach for all. So with that, I'll conclude. Thank you for attending and happy to answer any.
Well, thank you, Eugenio and Jeffrey, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here is for you, Jeffrey, and this question asks, despite the advancement around precision oncology, some argue its low conversion rate for patient trials based on genetic analysis, 4 to 11% for new drugs, about 58% for off-label use. They claim it isn't standard care, with many patients opting out or excluded throughout the process. Considering OmniSeq's insights, quick results, and targeted trial suggestions, do you notice increased retention in drug trials and in this pattern improving over time?
Yeah, that's a great question, and it could take me some time to answer that fully. And unfortunately, to answer it completely, we'd have to have access to the clinical decisions the provider makes, based on the report, and then the patient willingness to enroll in a trial. But and also given that testing is often ordered by non-treating physicians, such as pathologists, access to that treating physician and the patient's decision is really challenging. But things that we've tried to do to overcome this, and to make progress with trial recruitment, is to engage with pharma. We have lab alert programs where we send physician letters alerting them of a patient with a rare disease or a rare variant and a trial that they could be eligible for. And we also participate as a testing laboratory in support of clinical trials where the alterations and reports are customized for the markers required for that trial and for the patient eligibility. And then we could have a custom report where it's just synthesized down and filtered down to those markers so the site PIs can make quick decisions. So we're working with pharma on trying to get patients better access. We also have set up a patient registry, I should mention, called Prefer, which is in collaboration with CCORN, which is a community clinical oncology research network to understand impact of disparities in precision medicine for cancer patients. And this is trying to address, the underserved populations and regions, again, to improve access and diversity in clinical trials. So I think through these mechanisms, we're able to do a better job of getting the information across to physicians, but there's more education that has to be done. And we hope that, we can help through that process get higher trial enrollments, because not only to support drug development, but we also want to give, patients access to these emerging therapies, which might be really their only hope.
Great, thank you. Another question for you here. What are the next steps for LabCorp to continue improving the ability to provide patients with the fastest and most reliable placement into appropriate targeted therapies?
Yeah, another good question. And although I focused this talk on solid tumors and specifically tissue-based testing, liquid biopsy or blood-based testing is another option for assessing somatic alterations. Though not as universally established today as tissue profiling, the liquid biopsy has the advantages of being non-invasive. It can overcome some of the tissue limitations that we discussed earlier today. It can be used also for things such as therapy selection, determining tumor of origin. It can be used for early detection, response after treatment, monitoring disease over time. So it has a lot of applications. And we recently launched the LabCorp Plasma Focus Test, which is an actionable liquid biopsy assay, focusing on, again, on-label and guideline-driven biomarkers. But we're always enhancing the OmniSeq Insight tissue-based assay that was discussed today. We're validating and turning on new markers with therapy associations all the time, and always testing out system and process improvements that we hope we could continually reduce turnaround time, improve performance overall of the assay, and with the goal to extract as much information from these tumors as we can.
Great, thank you, Jeffrey. And it looks like we have time for only one more question today, and I want to make sure this one gets answered. It asks, will we be able to access the slides from this presentation afterwards? I would love to be able to look further into some of the papers that Dr. Conroy has cited. So this webinar will be available for on-demand viewing immediately after the live broadcast today, and you will be able to just jump back into this link the same way you did today and rewatch the presentation at any time. Jeffrey has also mentioned to us that you may request the slide deck to him if you'd like to reach out via email or ask a question in this Q&A box during that on-demand period, and he'll be happy to assist you there. So thank you both, Eugenio and Jeffrey, for your time today. Do you have any final comments for our audience?
Yeah, thank you for listening in. I hope that this was great experience as well as it has been for me to collect all this information and always trying to make better precision medicine. Thank you for listening.
Wonderful. Well, great. Thank you again, Eugenio and Jeffrey, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Covaris, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and no submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.